Welcome back. In this section, we're going to learn about semi-permeable membranes and the movement of materials. So let me explain what I mean by that word semi-permeable, because I'm not sure I covered it already. Remember when we were talking about the cell membrane, I said certain things like oxygen and carbon dioxide could easily pass, and yet other things were restricted in their movement. They maybe needed a channel or a carrier protein or couldn't get through at all. That's what it means. It's semi-permeable. It's selectively permeable as to what can get through. So when we're talking about membranes that are selectively permeable, we can talk about the process of diffusion. So here is an example of a selectively permeable membrane. And we have molecules, in this case, they're labeled molecules of dye on one side, and the other side just has water. And so what is happening is the dye is moving from the side of very high concentration to the side where there wasn't much dye. And they are going to keep on moving until the concentration is even between the two sides. And it's going to reach equilibrium. At that point, it doesn't quit moving. They don't quit moving. What happens is the number of molecules moving from this side to there is equal to the number of molecules moving from that side to this side. So it is in equilibrium as far as movement goes. And moving from a side of high concentration to an area of low concentration is called going down the concentration gradient. It does not require any energy and will happen spontaneously. Now, if we are talking about the movement of water, instead of saying diffusion of water, we're just going to use the word osmosis. So this diffusion that I just talked about is one of the examples of what is known as passive transport. Passive transport means no energy is needed. So in diffusion, we went from an area of high concentration across a selectively permeable or semi-permeable membrane into an area of low concentration with no energy. Another type of passive transport would be one where, for instance, we need a carrier protein or a channel protein, as seen in these drawings. We're still going from higher concentration to lower concentration, but these carriers or channels are facilitating the diffusion. So this type of passive transport is known as facilitated diffusion. So there's two types of passive transportation, just normal diffusion and facilitated diffusion. Neither requires energy. Both are going down the concentration gradient. Simple diffusion does not need a transport protein. Facilitated diffusion does need a transport protein. Now, what happens if you want to go up a concentration gradient? Like envision yourself like you're at a concert and it's very crowded up there. You're at the doorway. You just want to push and shove your way in. Okay. Do you see how you're going to have to push and shove? It's going to take energy to get in. So that is going to be called active transport. So look here, look at these diamonds. Okay. We've got 2 diamonds here on the outside. We've got 5 on the inside. In order to move in, it's going to take energy. It's still going to take that transport protein. So we don't lose anything from before. We're just adding one more thing. So we still need our transport protein. Now we're going against the concentration gradient, and that is going to require energy. So in synopsis, passive transport, super easy, goes down a concentration gradient, no energy required. 2 types. Active transport, this course, only 1 type. Although in reality, if you ever take human physiology, you'll learn that there's 2 types. but requires the transport protein and goes against the concentration gradient. So it requires a source of energy. Okay, so that's great for little things, but what if it's something big? What if that cell wants more than just like a little tiny molecule? What if that white blood cell wants to engulf a bacteria? How is it going to get it across the plasma membrane? Well, or how is it going to get rid of a big waste product? Well, there's two other processes known as endocytosis and exocytosis. So let's look at those words. These are the words for movement of larger substances. Cyto means cell. Exo means exiting the cell. Exocytosis, exiting the cell. So by process of elimination, endocytosis means entering the cell. So when it comes for stuff entering the cell, just think the cell is going to be eating and drinking, just like humans, we have eating and drinking. So just like humans, we've got two different words, right? Eating and drinking. So for the cell, we're going to have two different words, one for when the cell eats something solid, and one for when the cell drinks something liquid. So if the cell is going to have endocytosis of something that is solid, we are going to use the word phagocytosis because the prefix phago means to eat. So phagocytosis is one of the two types of endocytosis. So here is a cell with a solid particle, could be a little E. coli. Notice what happens is the plasma membrane of the cell goes up and completely surrounds it and engulfs it. And it's going to form a little vesicle surrounding that little piece of food. Okay. Now, here's a little watery environment. Say it wants some water, and maybe it's going to take a little bit of nutrients in with the water. It's going to do the same thing. It's going to surround some liquid. We're going to have plasma membranes surrounding it. So we're going to have another little vesicle surrounding, this time found in liquid, only instead of calling it phagocytosis, which means to eat, we are going to take the prefix pino, which means to drink. So think of like a wino drinking wine. In this case, it's pino. Okay. So pinocytosis and phagocytosis are the two types of endocytosis, how a cell takes in large substances. Now, for getting rid of large substances, there's only one method that's called exocytosis. It's exactly the opposite. We're going to have a little vesicle filled with the waste products that's going to go close to the surface. The cell membrane, the plasma membrane is going to fuse with the membrane of the vesicle. And it's kind of like popping a zip. And now the stuff that was inside that vesicle would be in the extracellular space. Okay. All of these processes, whether it's phagocytosis or pinocytosis, so all types of endocytosis as well as exocytosis are going to require ATP. This is work. This is the cell working. Okay. The next topic is something known as tonicity. If we take cells and we put them in solution, we want to put them in a solution that is what we call isotonic, as in the same tonicity. What that means is it has the same amount of solute in the solution, the same amount of dissolved particles in the solution as we have in the cell. So we have the same concentration of water outside the cell as we have inside the cell. So what that means is the same amount of water will go inside the cell as would leave the cell. So the cell is kumbayaing. It is happy. It's not going to shrink inside. It's not going to get big inside. It's going to stay the same size because there's no net water movement. This is very important clinically when doctors write prescriptions for intravenous fluids that we write overall for isotonic solutions. Now, if we take those same cells and we put it in a hypotonic solution, then the solution has less dissolved solutes, which means that solution has a higher concentration of water. So there's more water outside the cell and less dissolved particles compared to inside the cell. So if there's extra water outside the cell compared to inside the cell, that means that extra water wants to move inside the cell. So the extra water is going to go inside the cell, and that cell is going to blow up and up and up and up until it bursts like a balloon, and that cell dies. So a hypotonic solution is not good. So giving patients an IV that's just water or just sugar water for prolonged periods of time, that's not isotonic. That's not good. Similarly, but the opposite, if we take cells and we put them in a hypertonic solution, meaning there's more dissolved solutes in the solution. Say, for instance, we put it in a heavy salt solution. Say salt water, seawater. That would mean there's a higher concentration of salt in the solution than there is the concentration of salt inside the cell, meaning the extra water is inside the cell. So what does that mean? That means the water is going to be leaving the cell. And the cell is going to shrink down and shrink down and shrink down. And guess what the cell is going to do? It's also going to die. So that's another way of causing death. So it's very important not to give hypertonic IV solutions to patients as well. So yes, there is an online activity that can help you understand this more. So make sure you do the online activity I give for this. So the last thing I would like you to do for this section, I would like you to practice doing a compare and contrast, because this is great activities for you doing when you're looking at things in science in this course, like when we're talking about ways that bone can be made and other things we look at in this course. And for this one, I'm giving you a word bank, and I want you to do a compare and contrast for passive transport and active transport. And thank you so much for all your hard work in this class, and I will see you. And finally, we will go back and we will look at other things that are inside the cell and what they have to do with cellular function. I'll see you shortly.